Hello, good morning, good afternoon, good evening. Thank you for joining our International Plant Breeding Seminar Series. Just before I introduce Dr. Rustgi, I want to say that the first seminar of the series last Thursday, we have connectivity issues, and but the presentation was recorded by Dr. Rohayu Arub, and it's already part of the announcements. If you scroll down, every time you receive announcement, you see the recording for that seminar, and next time you will see it for this seminar. So again, thank you for joining. Today we have the pleasure to have Dr. Sachin Rustgi join us from Clemson University. He's an associate professor of molecular breeding in the Department of Plant and Environmental Science at Clemson. And he also serves as a faculty scholar at the Clemson University School of Health Research and the Center for Human Genetics. And he's also an adjunct associate professor at other universities. And his research focuses on developing dietary therapies for individuals with celiac disease or wheat and peanut allergies, identifying biomarkers for heat tolerance in soybeans and peanuts, and creating strategies to combat major insect pests and fungal pathogens. Dr. Ruski has published a lot of papers and books, and his academic excellence has been recognized in several locations. He's going to talk today about lowering immunogenic proteins in wheat and peanuts and toward a safer foods for sensitive individuals. Sachin, it's a pleasure to have you with us. I'm going to stop sharing here and give you the floor for you to share. Okay.
Sure. Thank you. Thank you so much for the kind introduction and also like for the invitation. Um, Here we go. Today's topic is I will just focus on like our research on the development of reduced immunogenicity wheat and peanuts. So we were actually working on this project from quite some time. It's about I will say like 20 years of research that went into this. So I will just first introduce you to the subject. So the food reactions to the food are multiple different kinds. They could be like the immune mediated and non-immune mediated reactions. So immune mediated reactions are the ones that are actually like a very high prevalence in the population, and you can see like these immune mediated reactions could be also of multiple kinds. There could be the immune reactions that are immunoglobulin E driven, like these, which are the primary antibodies produced against the food allergens in our body. And then there could be other reactions which will be actually mediated by the immune cells in immunoglobulin G, which are also the secondary antibodies. So these immunoglobulin E mediated reactions are instant. So these are generally the food allergies. So you will experience them right away, like as you consume the food, like within an hour duration, like you will start experiencing symptoms. While like the immunoglobulin G driven reactions are chronic. So these are the reactions where not necessarily the antibodies are developed against the allergens in the food, but sometimes against the enzymes in the body, like which are the metabolic enzymes needed for the body's housekeeping. So those are generally the food sensitivities which are celiac disease. So oftentimes people are confused between the two kind of reactions. I would like to emphasize here we will be talking about both of these reactions, which are allergies and food sensitivities like celiac disease. The other kind of reactions, as I mentioned to you, there could be like non-immune mediated, and these are actually lacking a certain kind of enzymes in the body to process the proteins, and many other metabolites in the plants. So specifically, like I would like to emphasize here, like the lactose intolerance is like the kind of reaction where one lacks an enzyme that allows digestion of the lactose in the body. So that those reactions are the intolerances. So do not confuse between the intolerance, sensitivity, and allergy. With that introduction, I will move on. So I will take like the first topic, like that will be like the gluten syndrome, like which I call, like there are multiple different kind of reactions observed on consumption of the gluten proteins. The gluten are the proteins in the wheat grain, and wheat endosperm, like which is largely the dead tissue, like that actually feeds the embryo while it's germinating, produce these gluten proteins or accumulate these gluten proteins. So this reaction to the gluten proteins are generally divided into three parts, like which is exposure to the wheat gluten proteins, which is environmental exposure. The second one actually has to have like a leaky intestine. So those individuals who are sensitive generally have a leaky intestine. So the tight junctions are actually not so tight, as you can see with my cursor, and then it leaks the undigested or not completely digested peptides in the bloodstream, and they were actually taken by the immune cells and presented, and then actually the antibodies develop against these cells, and they could actually lead to the reactions which could be the flattening of villi in the intestine. Like so, these are a chronic reaction which takes over like a course of time. So the reactions I mentioned is like the allergies, like the wheat, which are also quite prevalent. It's for about 0.5% of the population in the United States are affected by the wheat allergies. And there could be like other sort of reactions which are non-intestinal reactions, which are known as like the non-autoimmune reactions, like which these reactions are actually like less well understood. Like these are generally called like wheat sensitivities, or um, earlier it was called like the gluten sensitivity. Now we know like it's not necessarily because of the gluten alone. Like there are also FODMAPs in the grains, which are the sugars like in the grain, like which could lead to this reaction. So let's move on. So I would like to first introduce you to the wheat, like the people who do not work on the wheat. Like the wheat is a very complex organism. Like so, it actually evolved from the two successive rounds of hybridization that took place in nature. So one took place about 5.5 million years ago, when like the A and B subgenome gave rise to a tetraploid wheat in the Middle East, and then it actually about 10,000 years ago it actually produced the hexaploid wheat by actually having another round of hybridization with Eglops torsi, like Orchiticum torsi. And it gave rise to the bread wheat. So bread wheat is, as I mentioned, is extremely complex. It consists of like the three subgenomes, which we call A, B, and T. And wheat genome, you can imagine it's extremely large. It's about 17 GBs in size, which is 5 times of the human genome. It actually, each of these chromosomes are actually much larger than the rice genome itself. Like, so the smallest chromosome in wheat is actually larger than the total rice genome. You can imagine the complexity. And then these loci that actually code for the gluten protein, there are 12 loci known with 97 genes, which are distributed over like six chromosomes belonging to two homologous chromosome groups. So it's a complex of like multiple loci, and then each loci having multiple genes. So there, the number of the genes varies from 6 to 44 at each locus. With this introduction, I would now show you like, I mean, the gluten proteins. So the gluten proteins are actually are consist of like two different parts. Like, so there are monomeric or monomeric proteins, which we call gliadins, which are generally globular in structure except for the omega gliadins, and they have like they are cysteine rich, and except for the omega gliadins, which are cysteine poor. Like they make like the globular structures and are monomeric. And then the other kind of the proteins are actually the polymeric, and they were resolved on different kind of gels. So you can resolve like the gliadins on the acid page or aciduria page, and then the glutenins on the SDS, sodium 2-decyl based polyacrylamide gels. And they were also extracted using the differential extraction method that was actually perfected in the 20th century, like early 20th century, by American scientists. These proteins, when resolved on like a two-dimensional page, you can say that each wheat genotype could produce from somewhere from 50 to 100 proteins. So it's a very complex mix of the proteins, where the gliadins provide like the viscosity to the dough, and the glutenins like provide like the elasticity, and together they provide the viscoelasticity which is needed for the baking properties. As you can see, like it produces a airy um, crumb which trap like the air, like, and it actually produce like this neat bread structure that we all love. So now I will go into like the strategies that we have taken like to deal with this gluten syndrome, or like you can call like the celiac disease. So we have taken like the two approaches, like to eliminate gluten, like one is epigenetic and the another one is like a genetic approach by targeting the actual genes coding for the gliadins and glutenins. So epigenetic approach is actually based on like this knowledge which came from the barley research, like where the proteins which, like say I mentioned you, the gliadins and glutenins, the promoters of the gliadins and low molecular weight glutenins are actually like methylated and demethylated. So they are controlled by methylation in the endosperm tissues, and the methylation has to be removed for their expression in the endosperm. While they continue to stay methylated in the vegetative tissues like leaf, while the promoters of the high molecular weight glutenins are largely controlled by the transcription factors and repressors, and they are protected for methylation, demethylation with the presence of the CpG islands in them. So there are two genes like which we call like the Demeter, which is the glycosylase, which remove the methyl groups from the promoters, and there is another protein that is called like the DRE2, which actually installs a iron sulfur cluster on the top of this enzyme to make it a holo enzyme and functional. So to prove the function of the Demeter gene, we first clone it about like more about like I will say 15 years ago, like, and then after like cloning, we actually developed a hairpin construct which consists of the 185 base pair stem and 500 base pair loop, and we attach that hairpin with like a high molecular weight gluten promoter, so it will only express in the endosperm. Because we have actually figured out like early on, like when we create mutations in the Demeter gene, it actually leads to pollen sterility. So we introduced that using the biologistics approach, and then we screened these putative transgenics. So we had like about 118 of them, and out of those, like about like 7 lines actually showed like the kind of inherited the transgene perfectly, and then showed reductions in the gluten patterns. We have published that long time ago, like, and then you can see the results. Like those transgenic lines actually were reduced in the content of these gliadins, as has been shown by the HPLC and also with the PHL. While as these hairpin constructs could lead to off targeting because they are pretty large arms and they could be processed multiple different ways, like and produce multiple siRNAs. So we decided to actually produce artificial microRNAs using the rice microRNA 528 as a template. We designed two microRNAs, one from the active site, another one from the internal region of the gene, the Demeter gene, and we attach these artificial microRNAs again with the hemagglutinin gluten promoter introduced using biolistics approach to the plants. We had about 213 candidate transgenics which we screened, and then we found like about 12 of these inherited the transgene, and then when profiled with the protein, you can see like in comparison to a just bar transformed line, they had significant reductions in the gliadins. So we noticed like they have reductions up to 89% of the reduction in the gliadin content. While it's not perfect, like, but it is significantly low and could lead to like a useful product. But as we are all aware, like the transgenics are yet not accepted except for like a few cases where the government approval has came. And I would wish to emphasize like the approval and acceptance are the two different things, which I will come back on this matter at the last slide. Okay, moving on. We also decided to actually improve the Demeter silencing at two different levels. So you see like we have created the post-transcriptional suppression, but we could actually have the post-translational elimination by actually getting rid of the DRE2 enzyme. As I mentioned, like it actually installs the iron sulfur cluster and make it a holo enzyme. So to deliver these constructs, we took help like it was done like at the time like when these techniques were developing for site-specific mutagenesis. Like, so we took help of like the tail repressor for Demeter gene, and then we thought like we can actually introduce that by insertional mutagenesis into DRE2 gene using the Cas9. So we developed two constructs called 7-3 and 24-3, and we delivered it using a pull-in magnetofaction method that was also coming up like at the time, and later proven like to be successful in corn, like, and then in cotton. While there were a lot of rumors, like in the meantime, like there were reports which said like the polymagnetic fiction doesn't work, like, but it actually worked in our hands. So I will just go over like the method, like, so this method start with like collecting the spikes at the Ficki's developmental stage 10 and 10.2. and then you expose the spikes like after removing or trimming the awns like to the light, so the anthers protrude out like, and the anthers were collected. The anthers were ground in the pollen medium, and then the pollens were extracted, and the pollens were exposed to the plasmid DNA coated onto the magnetic nanoparticles, which are about 200 nanometers in size, which can traverse through the pollen, the pore in the pollens, like with the help of the magnetic fields. And after that, like these pollens were evaluated for their viability, and they were actually the key point I would like to emphasize is like the pollens were dried before they were delivered to the plant. And this was mostly like a cross pollination, but like a kind of akin to a self pollination, because we took the pollen from the same plant, modified it, and delivered on the back onto the plant. So we did two kind of tests. We did like the pollen viability test using also pollen germination, and then we delivered the pollen to the spikes, and then we harvested the F1 seed, and then F1 was also like, I mean, grown into the greenhouse and evaluated. So before doing like this, we actually tried to figure out a ratio between the magnetic nanoparticles in the plasmid DNA to figure out the carrying capacity of these magnetic nanoparticles. We turned out 1:3 proportion of like the magnetic nanoparticles to the plasmid DNA is a good carrying capacity for those magnetic nanoparticles. So we use the gel retardation assays for that purpose. You can see like, I mean, if you see all the DNA in the well, like that's retarded with the kind of the magnetic nanoparticles. So these magnetic nanoparticles were also evaluated for their size, like after treating with polyethylene imine, and then their zeta potential, so we can load like the DNA onto it. Like, and these were tested first for delivering a um, GFP protein, the GFP construct into the pollen grains, which was driven by ZMA's promoter. Like, so it was driven into the pollens, and then it actually produced like the transiently like the GFP. So it kind of proved like first like the method, and then we delivered it like as the method I have shown you in the previous slide. We grown like these Avon plants, and then in the greenhouse we collected the leaves and we tested it for like the two constructs. So 7-3, we saw like about 7% of the plants were positive with the constructed specific primates. We also sequenced and confirmed the product. And then later we also tested for like the 24-3, which is another construct, like about 36.9% of the plants actually were positive for it. And then we screened like the F2 progeny of some of the positive plants. About 5% of those F2s with the nested PCR actually showed positivity. So that actually kind of showed like this method works in wheat. And then later we these plants that were positive were also screened for the mutations in the DRE2 region. Like, so far we have not found any insertional mutagenesis, but we found like the point mutations in the DRE2 gene, like which were actually substitutions in small indels, which were tested using the next generation sequencing approach, as you can see here, and then were also confirmed using Singer sequencing. So, in addition, like to the kind of the cis and trans regulators that were pre-identified, like say Dimeter and DRE2, like we also started to look for the novel epistatic regulators of these gluten proteins. For that purpose, we actually have our hands on like this DNM population, which is developed by Eric Wilson in the Michigan State University, and it was actually a cross between hexaploid wheat and seven different donors, sorry, eight different donors of like the D genome, because as we have shown, like it has been shown before, like the D genome carries the heaviest load of the epitope. So we thought we can actually introduce more alleles in the D genome, and hopefully will have like some reduction in the epitope content, as well as found like the novel regulators that can actually regulate like the the gluten protein accumulation. For that purpose, we did like the phenotyping of the gliadin gels in two different ways. Like, so one way was like to record like the presence and absence of the gliadin bands using a gliadin reference, which is kind of a pan gliadin reference that we have created using all the lines for this. It's about 421 lines of integration lines of this population. So as you can see, like it actually lead to like this protein phenotype that we use eventually for the mapping purpose. So we did the GWAS analysis using the SIP data that we have obtained from Eric Olson, and then once we did like the mapping, we found like the signals, 11 signals, which were actually detected on all except for one, that is the 4D chromosome of the D subgenome. And these signals were actually confirmed by many different, they were detected by many different analytical methods. So they were actually carry some confidence in them. As you can see, like there is a signal detected on the chromosome 7DS on the short arm, like which was actually kind of, there were two signals, but they were very close to each other, like which were actually kind of for all three kind of the gliadins. So we use this information to develop the gene interaction networks, and then we notice like there are both kind of regulation cis and trans for like the gluten genes. um, and the most interesting thing that we notice is like that the regulation goes beyond this D genome, like so it's the D genome is regulating the proteins encoded by the A and B subgenome, as you can see in this interaction network. So when we look like at the literature, so there is a lot of genes that were regulating the gluten proteins were already mapped onto the chromosomes. So when we reviewed, like there were structural genes as well as there were regulatory genes that were actually mapped on the D subgenome, and we thought they could serve as kind of the candidates for these QTLs. I have actually pointed out some of these genes which were highly interested. Some were actually the structural genes for the gluten proteins, which is on group the 1D and 6D chromosomes, as well as there were actually transcription factors that are of high interest, which binds to the prolamine box. As you can see, the interacting regulatory network worked out for these proteins. So you can see their promoters consist of the core promoter, which is like proximal to the start transcription start site, and the distal promoter, and they were actually controlled by methylation, demethylation as well, like as I mentioned earlier to you. So we have worked on three of these proteins, and I will show you the results in the coming slide. So we, as I mentioned, that we have worked on these prolamine box binding factor as well as like the steroid protein activator. These are two transcription factors that bind to the prolamine box of the gliadin and low molecular weight glutenins. So when we looked at the single mutations in the chronos background, so chronos is a tetraploid beat and cadence as a hexaploid beat. So we obtained the single mutants in A, B, and D subgenomes. We already saw reduction in the content, specific eliminations of the gliadin proteins, as you can see in the highlighted lines. I have highlighted them with the red and blue colors, and you can see the specific gliadin proteins disappear in these single mutants. And the idea is to pyramid them by actually making crosses and then eventually make these crosses with the structural gene defects that I will show you in a minute, and then create a leak-proof mutant. So in addition to these mutants, we also looked at the mutations in the structural genes of the gluten proteins, and that was actually done through like screening the wheat land traces. Some were already recorded in the past, but their purity has been lost over the time. So we have screened those lines, and we have obtained the mutations that were the single mutations actually bred into the Picasso backbone. So this is a wheat variety called Picasso, where introgression lines, or like I will say the near isogenic lines, were created for single mutants, and later they were crossed to create double mutants. So you can see and we compared that with the wheat diploid progenitors. So their proteins were extracted and then resolved onto the aciduria page, and then we made medleys of these proteins. The idea was to actually assign bands to specific genomes and subgenomes, as well as like to the chromosomes, and then and confirm like these mutants, which are the near isogenic lines in the Pegaso, like to be pure, and then carrying those mutations, as well as characterize the bends in the land traces, as has been shown here. So in this slide, I'm showing like little bit more details of some of the land races, that is Clements, Seri, and Jamir. Like we evaluated them by multiple different methods. Like so, these methods included resolving them in the page L. As you can see, like there were actually missing bends for the omega gliadins, gamma gliadins, as well as alpha beta gliadins. We also did like the gluten-seq, which is a next generation sequencing method, selective sequencing method for the gluten proteins, developed in 2019 by a Spanish scientist. Um, uh, and then we also did use the chromatography, and the chromatography also proves Clement to be actually lacking these omega gliadins. So this was a run for only the omega 5 gliadins. And then we used some gliadin specific DNA markers that were previously developed. And then you can see in these gluten seq coverage plots, there are mutations on the chromosome 1b and chromosome 1a. And as I have shown you, in the previous gel, we tried to map the protein band, so it kind of tallied with the results of the proteins and the DNA. So we kind of was able to characterize these land races much better and could be used for breeding purposes in the future. So the Picasso mutants were also evaluated using the chromatographic methods. So these were the single and double mutants, and the chromatography has also confirmed the loss of those specific proteins. You can see the chromatography is based on the hydrophobic properties of these proteins. So the different methods have actually evaluate these proteins in different ways, but they confirmed and reconfirmed the lack of these. So this was a slide showing the mass spectrometric analysis of these single and double mutants, and you can see it reconfirmed the idea of these lines. So all of this was actually before going for more intense breeding effort that we have done here at Clemson. So, as I mentioned to you, we also have access to a triple mutant, which is a omega gliadin free mutant. As you can see, it was lacking gly 1A, 1B, and 1D. So, three loci, which are coding for omega gliadins, were actually lost in this mutant. So, this is the triple mutant for omega gliadins, which was crossed with a double mutant, which is a 6A. So, gly 2 6A 6D null mutant. So to create a pentaploid mutant so pentapole mutant lacking all five of these gliadins and result into this kind of profile. So for that purpose, we made these crosses. We dissected the grain into the embryo half and the endosperm half. The embryo half was used to produce the plant, and the endosperm half was used for gliadin profiling using the page gels as well as liquid chromatography. The whole idea is actually tested using the T cell assays. So we already have access to the two different T cell lines derived from the peripheral blood of the celiac patients, and then study them for processing quality. That's up in air yet. So we have characterized those F2 and F3 lines that we have created by crossing those two mutants that I've shown you a minute before. So we have resolved these on the gliadin proteins specifically onto the aciduria page. As you can see, they were actually lacking specific proteins, and we were able to actually obtain up to a pentapole mutant, which was, we believe, it is a homozygote for four sites and a heterozygote for the other site. We have also done the profiling using the gluten seq, as I have mentioned to you. It's a selective sequencing method, and you can see the different peaks were actually highlighting those gluten genes, and their coverage was lost in those mutants or the cross material. And we have actually confirmed some of this using the PCA-based markers, which were developed for selected genes. The boxed areas were actually where we have tested the markers, so it kind of validated those deletions as I have been shown in this cartoon. So this was actually more of a repetition of what I have just mentioned to you. It is a uh, uh, full blow of the sites using a heat map. We were showing the loss of coverage in those. You can see the Pegaso single mutants were for 1A was lacking 1A. 1B was lacking 1B site, and D was lacking 1D site, and then A and D combination lacked A and D, BD combination lacked B and D, and then this is a triple mutant that we use as a parent, which was lacking B, A, B, and D, but there is actually a slight loss on the A in this particular case. And then this is our F2s and F3 lines that you can see where my cursor is. And then these were two controls. So it's Vesper 926 is a spring wheat variety, and the Picasso was also used um, as a control in this case. And then this Saint Pastor is one of the land races which we kind of confirmed it carries a deletion in the Gly 1D. So this is continuation of the previous slide, which shows the results of the Gly F2, like which is for alpha beta gliadins from group 6 chromosomes, and 6A and 6D sites. You can see like the double mutant carries shows lack of coverage in this area, and our F3 and F2 lines also have like results. So F2 lines were segregating, that's why you were seeing it actually had like the D, but we found a segregant in the F3 which was actually lacking both of these. We also evaluated like the epitope load of these lines by actually evaluating those sequences from the gluten seq, and then we noticed like the arrow head was actually showing the results of the F3 line, which was actually showing significant reduction in the epitope load. Based on this, we have also conducted the enzyme linked immunosorbent assay like using the R5 antibodies. I would like to emphasize here these R5 antibodies are actually like can detect is developed against the omega secalin from rye, like so it's actually, but it can detect all gliadins, so it's like cumulative gliadin quantification using this antibody test. And as I'm reporting here, like these F2s and F3s actually have significant reductions in comparison to the wild types. So the next thing that we have done in this area like was actually to use the multi-gene editing approach. As you know, like I have mentioned to you, like these proteins were coded by um, six different sites on like six different chromosomes. We aligned the sequences from and there were multiple genes at each site as has been shown here. We obtained their sequences, aligned them, found the conserved sites for guide RNA development. So we developed 2 to 3 guide RNAs per site. We tested these guide RNAs in vitro using two different methods, a gel-based assay and then also a chip-based assay, like so, which actually shows difference in the electrical conductivity. These after testing these guides, we assembled a construct. Um, and that construct was developed using the construct developed in the University of Maryland as a template, and then we introduced them using the biolistics method to the embryos that were derived from the mature grains. Pay attention, it was done on the mature grain derived embryos, not from the mature grains. We screened these lines for like the T0 generation and T1 generation for the positives for the Cas12a, and then also looked at the mutations at the target site in omega and gamma alpha and gamma gliadins. The results of the omega and gamma alpha gliadins are shown here, like so it carry deletions of say like from 12 to 5 nucleotides large, like which were actually very likely to happen with the CPF1, which actually is known to cause deletions of that size. So now I will move on to the next problem that we were dealing, like which is the peanut allergies. So as you can see, the peanut is produced in certain sites in the world. Like as you can see, they were highlighted in the green, like but it's consumed even further where it is produced. And the food allergies also exist almost all of the Western world except for like the say the global South. Like so and the reason for that is not fully understood, like but what we understand today is like the early introduction of the peanuts actually helped develop tolerance for the peanuts. That's why like the allergies were not detected in the global South where the introductions were quite early, while these instructions have been followed in the global North as well, like now, and then there were benefits reported very recently, um, just like a couple days ago. So peanut allergies have increased in number, like from 1997 it was about like 0.4% of the population. Now it was actually 1.8% of the population that is affected. About 16 proteins in the peanut grain actually cause these peanut allergies. Among them, four are major allergens, which actually are kind of like which can cause allergenicity in like more than 90% of the individuals who are sensitive to peanuts. Like so they are quite allergic, like so the remedy at the moment is like to avoid peanuts. And there were two other therapies that are available, like one is known as Xolair, that is actually more of like a targeting the immunoglobulin E that actually binds to food allergens, and it's non-specific to like peanuts. Like it could be used for any food allergies, like and then there is another one that is oral immunotherapy that is known as Velforsia, that's actually introducing peanut proteins in small doses, major doses to the patients over the course of time so they can develop tolerance. So in line, these or OIT is what like we wish to actually create, like in our case as well. Like, so I would like to emphasize here, like those oral immunotherapies are quite effective, but they are pretty expensive. Like, so it costs like about $60,000 per annum per individual. Like, and then say Xolair, which was like another therapy that I mentioned to you, like costs about $30,000 per annum per individual. So it's pretty expensive. So we thought like we can develop some uh, affordable therapies like for the period sensitive individuals. So we took two approaches here, like germplasm screening and genome editing. The idea was to actually develop the molecular markers that allow the breeding for this trait, as well as like to develop the multi-gene editing platform that could be used for this purpose as well as the other purposes in peanut world. So we screened like the large germplasm collection. So we screened the peanut weaning core collection from the United States as well as the collection from South Asia. And then there is a ICRISAT mini core collection, which is the International Crop Research Institute, um, which actually has a very large collection of peanuts. Like, so we screened those lines using three different methods. Like, one is like running them the proteins on the SDS-based gels, which is polypeptide gel, and then followed by densitometric analysis, enzyme linked immunosorbent assay, and chromatography. And those all three methods actually kind of showed um, some sort of correspondence, as you can see in the next slide as well. So we use this information to map the sites, like, or use for GWAS analysis to identify the cis and trans regulators of these proteins. As you can see in this, like a plot, like there were the high accumulators and the low accumulators. I would also like to emphasize here, like there is lines which we call low accumulators, not necessarily be actually low in all four proteins like that we were targeting. That these are the four major allergens that we were targeting via the densitometry or enzyme linked immunosorbent assay. So we have narrowed down the samples. We did like the densitometry and ELISA for all lines, and then we narrowed down it for the liquid chromatography and then liquid chromatography and mass spectrometry. So we used like the densitometry and ELISA data for GWAS analysis, like, and we found signals on like almost all chromosomes of the peanut. Um, and then we noticed there were some, so I showed like the carrier chromosomes which were actually having the structural genes for these ara proteins with a red dot, like, and then you can see there were the MTAs identified, like for like both ELISA based and then densitometry based assays, and they were kind of sites where there were signals for multiple of these tests and multiple of these proteins, and they were of high importance. So not only we did a screening of these collections, we also screened like the diploid progenitors of the peanut, and then some other varieties that are commonly known, like say Gregory is a common variety, Virginia type variety that is used. As I would like to also mention here, like the peanuts have multiple market classes as the wheat does. Like, so peanut has like four different market classes known here. Like Virginia is the large bold peanuts, and then we have runner types which are very prevalent in prevalently grown like all over the United States. I will touch bases on this like in a later slide as well. Like, so, and then there are two small classes, Spanish and Valencia types. So we had like screened those materials as well, like for the ARA proteins, and then we were lucky to find like there is Arachis diagoi genotype GKP10602, like which was significantly reduced in the content of these ARA four major allergens. As you can see, the number one is Arachis diagoi on this gel map, like, and then we were lucky to have like a population was created in North Carolina State University, where the introgression lines, there were about 121 introgression lines of Diagoi into the Gregory background. So this population was screened for their proteins using different methods, and then were genotyped. And this information was used for the genetic mapping. Like, as you can see, like when we use like a Hudson alpha based sequencing based genotyping methods, we identified a signal on chromosome 4, which is a trans site for the ARA H1 and H3. And this signal actually overlapped, and 96 of the genes actually underlie this signal. And there were 8 genes that actually transcribe at the same time where the ARA genes transcribe, like, and they actually belong to transcription factors. They are interesting transcription factors. They are interesting candidates for this site. So not only from this analysis we identified like the the noble sites or regulators, we also identified the lines that we thought we can pyramid by genetic crossing. So that's what we did. These were the introgression lines that you are seeing. So we selected introgression lines, um, and then we made genetic crosses between them, and then we screened the F2 seed of these crosses, like using two different methods. The one was the gel-based assay, like densitometry, and then enzyme-linked immunosorbent assay. And then you can see the results of cross 1 and cross 4 showed like the pyramiding, successful pyramiding of the lines. Most of the seeds actually retained the size. You can see some shriveled grains, but that's not like we just showed them, but that's not like most of the grains look like. So not only from that analysis that we have done, like the diversity analysis, we identified this Arachis diagoi. We also identified another line called NC25. So uh, fortunately for us, like there was already a pre-existing population, North Carolina State University, which is the RIL population of 176 lines at the F7 generation. Like, so this was graciously given to us, like to analyze under a collaboration. So we screened this line again for these allergenic proteins, and I have emphasized here, like this line has been earlier identified by two different workers as well, like to be reduced in the content of these toxic or allergenic ARA proteins. So we did like the densitometric analysis of these lines. For We did like this analysis for year one and year two. Like, so we had seeds from 2019, 2020, 2021, sorry. Like, and then we have screened them, and then we identified high and low groups, like, so high accumulators and low accumulators of these lines. I would like to again emphasize, not all four major allergenic proteins were actually reduced in all in most lines. So we use that data for genetic mapping. Um, the results of the genetic mapping again found like the cis and trans sites. And there was like also a signal detected in chromosome 18, which is a cis site. So that means like where the structural gene for the ARA proteins exist. So it's a structural gene for ARA H2 and ARA6, and we found a signal for ARA6 on this site. Uh, Again, like we use these selected RALs for genetic crossing. So we made multiple crosses, and we are in the process of screening these crosses right now. We have also evaluated these lines using Western blotting with the help of our collaborators in the University of North Carolina. They were like the antibodies developed against these ARA proteins, and then we screened these populations. So this is just an example of like that. We have much more results than this, like what I have shown here. Like, but you can see like there were specific proteins lacking or reduced in like specific genotypes of both populations. And we have I'm also showing a plot of like a immunoglobulin E, which was hybridized like onto the Western blots, and then you can see the reduction of proteins like interaction with this, like, which is very encouraging. This actually allows us to actually identify lines for a more detailed test that I will show in a later slide. So other than that, much like wheat, we have also decided to use genome editing to do multi-gene editing of these genes. So we took the exact approach, like we identified sites on these ARA proteins, and then developed the guide RNAs, tested these guide RNAs using in vitro methods, which is like again gel-based and chip-based assays. And the guides that were actually showing good processing of the product, like, were actually assembled into two different constructs. So one construct was for bioelastic and agro-based delivery, and then the another one was actually using a novel method of like a virus-based delivery method. So I will show the results in a minute. So we first tested this construct, like, that was actually developed for the virus-based delivery, which carries also the GFP protein in it, using a protoplast transformation method. So we transform the protoplasts. The protoplasts are like the plant cells where you remove the cell wall and use the polyethylene glycol, like, to introduce the DNA into the cells. And then we monitored like the GFP expression, GFP protein. As you can see in the overlay, so red dots are showing like the chlorophyll, and then the green is showing like the GFP, and then the overlay where you see green is like the true GFP signal. And then we also sequence these to find mutations at the target sites in the ARA proteins. We have two target sites in RIS3, so that's why you are seeing two plots. Right here, we notice the deletions in these genes from somewhere ranging from 11 to 17 nucleotides. Um, and we confirm some of these results using the Singer sequencing. So after getting these encouraging results, we delivered this to the plant. This was the viral-based construct using a vacuum infiltration method where we delivered like the viral particles in the liquid to the plants under vacuum. And then we tested like the presence of the virus in the young leaves and the old leaves. So old leaves means like the leaves that were pre-existing the delivery into the plant, and the young leaves that formed after the delivery. And then we noticed in the young leaves the presence of a GFP specific product. We also monitored the GFP like yeah, in the xylem vessels like in the phloem, as you can see right here, like, and then we also sequenced the product to confirm like it's GFP. We also looked at the mutations at the target sites, and then we found like mutations. However, like we notice here, like the one difference, like the size of the deletions was relatively small in comparison to what we noticed in the other protoplasm-based method, and we don't know what is the reason behind it. We also delivered it like using the biolistics method, like to the chelide derived from the peanut seeds, and then and after delivery, like we screened the T0 plants for like the mutations at the target site. And then as you can see, like we noticed like the deletions in all four of the genes. Interestingly, there were more cases of like mutations in the genes that were actually at the periphery of this multi-gene editing construct. So we thought like maybe the processing of these guides have something to do. You can see like these guide RNAs were attached with the ribozyme, which is hammerhead and HDV. These ribozymes was something that was actually close to the ribozymes had processed better. So this is just our hypothesis. We don't know for sure, like, but we think that is the reason behind it, because there could be other reasons like the location of these genes, like methylation, demethylation, and condensation of the chromatin, like that could influence the number of mutations seen in these sites. So we screened these materials like at the T1 T2 seeds. We did like the same method. We cut the seed into embryo and endosperm half, sorry, cotyledonary half, and the cotyledonary half was used for the protein extraction, and the protein was resolved on the page shells. And you can see like there were families of the transgenic lines, putative transgenic lines, which were showing reduction in the proteins as well, like, and which corresponded with the mutation of the target sites. And these lines were also screened using the Western blots, as you can see right here, and the immunoglobulin E interaction. So there were some encouraging results observed here, like specifically I highlighted this 29-2 line, which is one of my favorites. So as I mentioned to you, like all of this material has been done through the human immunoglobulin E testing, and then this is a mix of results that I'm showing here, like which was including the material from the two populations that I have shown you, their parental genotypes, as well as the cross material and the transgenic lines. And I have highlighted some of the materials that were actually quite interesting, which showed the significant reduction in interaction with the human immunoglobulin E. As you can see, there were blanks in those lanes which were highlighted with a red dot. That's very encouraging, and that is kind of the kind of selection of the candidates for the next level of the testing using the basophil activation test. So this test is like kind of gold standard for testing like something for immunogenicity, immunogenic potential. So we this method, like I mean these basal cells like actually release the CD60, which is which you can see here, like when it's activated when the food allergen bind to these IgEs. So this test has been conducted like so this so far like we have done only with the one donor, like, and it was done at the University of North Carolina by our collaborators, which have screened like the five of the selected lines for that purpose. And then here you get like the blood samples, and the blood samples were tested with the media alone. That means like just the media, and then you have the the IgE derived from the patients, and then you will have like the food allergen that actually add to the thing. And we flow cytometry based method was used, which is known as flow gating method, like where you can actually flow all of these immune cells through the thing, and then there were fluorescent detectors that allow like finding you or gate these cells which you are interested in, like so that has been done. And then you will see like the activation in the samples where you introduce the food extracts. So if there is activation, how much activation has happened can be seen, like so there were different doses of the food allergen has been offered, like as you can see this line 571 showed like interesting results, which were all doses, like it actually showed low kind of induction in this particular case. And this is the area under the curve. This is kind of the same results shown in a different way, like, and you can see like with all doses, like this line pans out like to be interesting. So here I'm actually trying to show you like something more related to the breeding, as this breeding based symposium. So we have also evaluated the impact of the domestication while we were screening these materials. We looked at like the diploids and natural tetraploid and synthetic lines of these peanuts, and then we looked if the domestication has changed the content of the immunogenic proteins. So we have extracted the total protein, resolved it on page shells, did the densitometric analysis, and also studied the content of the immunogenic proteins, the four major allergens that we have mentioned earlier. We noticed no change in the content, as you can see down here in the plot, no change in the content of the immunogenic protein. So it's very clear the domestication has not increased the content of immunogenic proteins while the total protein content has changed. So we have also looked at the impact of the breeding over the years. Like so, we have screened the lines that were released in different decades, both runner kind and Virginia type. As I mentioned to you, there are four different market classes of this. Like runner kind is the most prevalent kind, followed by Virginia, Spanish, and Valencia. And they were. This is a map showing like the distribution where these lines are bred in the United States. And then you can see like over the decades, the protein content has increased in both runner kind and Virginia kind. So we were breeding for protein content, but we it has not led to the increase in the allergen content, as you can see here. Um, and then we also studied the impact of the heat stress and the protein content and composition, especially the protein composition. Like so, you can see like we were expecting the changes in the weather patterns. Like so, we thought we should also evaluate the impact of heat on the protein composition, because it's extremely important. If we release like these reduced allergen content line, what will happen when they were exposed to the future environments? So we did this like, and as you can see, the yield get a hit like when the temperature reaches like the 40-degree day and 30-degree night times. Like so we kind of designed the experiment with the help of our collaborator in the Clemson University, Dr. Narayanan. So we have performed these tests, and then this is the outcome of these lines. So there were the we have exposed the lines, the popular varieties of the peanuts, like to the optimal temperature and heat stress, and studied their protein profile before and after, or in the two conditions, sorry, the two treatments. And then we noticed like the heat stress generally reduce the contained immunogenic proteins. As you can see in these plots, there were some significant differences with certain lines. And then you can also notice like there is the reduced allergen content line has significantly reduced the content of the immunogenic proteins when they were exposed to the heat stress. While we also studied like some of the other material that we have access to, like through our collaborator, which is kind of a mapping population that has been screened earlier for the lipidomic content, like but they showed no change in either in the protein profile. So it's kind of clear there are multiple different mechanisms. Some of the lines actually play with the protein composition, and some with the lipid composition. And then this is the material that we have also screened, like for the pollen viability, the tolerant versus susceptible lines that we have see like, and we see no difference. So there are multiple different ways. Um, and this last slide, I would like to emphasize, like as I mentioned to you, like the acceptance and approval and acceptance are two different things. So we not only did like these experiments in plant breeding, we collaborated with a social scientist in the Temple University, and then she helped us run like these surveys, like on about 1100 individuals in each case, like where we kind of studied, um, kind of introduced like or the general public with like the two concepts and the existing therapies, like, and then we also tried to compare their opinion about like the materials developed using the conventional breeding approach as well as like the materials developed using genome editing approach. And you can see for the wheat, the outcome turned out like the people were more interested, like they thought like, okay, genome editing is morally acceptable, but they were not willing to actually intend to buy or consume these kind of grains in case of wheat. While in case of the peanuts, like their opinion differed. They were actually kind of willing to consume like any kind of the material developed. And we thought like it could be, there could be multiple reasons behind it, but we thought largely because the peanut is like more used as like as a luxury item, like over like the wheat is like kind of a staple. So people are more reserved, like actually accepting a genome edited wheat than like the peanuts. So with that, I would like to thank my collaborators, and then my past and present lab members as well as the funding agencies. Yeah, with that, I will
Well,
be.
thank you so much, Sachin. Very, very interesting research. I'm gonna abuse the floor for a short question. Um, have you tested the baking quality of some of these mutations in wheat?
Not yet of
Okay.
these mutants, but we have tested the baking property of the transgenic lines that we have created. And those transgenic lines, they were actually created in a soft white winter wheat. And as you know, the soft whites are
Yeah,
generally
yeah.
low in protein and they were used for cookie and have a soft dough. And they
Mhm.
it turned like the dough mixing properties changed to like a hard wheat and baked into a bread.
Oh, really?
Yeah, so it was very interesting.
Oh, the positive change, I would say. Yeah. So we have a few questions, and please pose the. I mean, we only have 5 minutes for question, but we can relay the questions to Dr. Rustgi later. He wants to know how long the genome editing processing we took, and additionally, how you managed to obtain the triple mutants across all the three genomes.
So yeah, so we not necessarily have triple mutants in case of like those genome edited materials in wheat. And then it took like us over 2 years, like so 1 year like to create the materials and another year to evaluate it. Um, and it's an ongoing process like to actually screen the later generations of it. And I think it's still like the breeding has to be done like to pyramid those mutations. So we didn't actually see like triple mutants in there.
Okay, and how long do you take to do the gene editing process from end to end?
It's in a single year.
Single year. Okay. Emma Brown commented that she really valued this research you're doing from the perspective of somebody who has gluten sensitivity. Okay, thank you. Um, Rachel said if you have a chance to bring all these materials to be tested in the field.
Yes. So the idea is like, yes, to test. So we were actually planning to actually test these materials like more. So right now it was mostly greenhouse confined. We have, however, tested some of the materials that we have created transgenically while back in Washington State when I was working there. So we tested for 3 years like in the field for the performance. We didn't
Hmm.
notice like any sort of like terrible susceptibility of the things, and same has been done like for the transgenic peanuts, not by us, but by the people in the University of Georgia. Dr. Peggy, like she has evaluated those materials that she has created using a transgenic method. And mind, like our method is gene editing, so it's non-transgenic. We
Yes, yes.
Yeah, so so she has done it like she didn't see any susceptibility for aspergillus. And I mean it's still like a lot more to be evaluated, and that will be a future plan. Yes.
Hmm. Okay. How much interest is from industry on this and the work you're doing, particularly in wheat?
The wheat, I have actually had like some interest early on, like when I was in back in Washington State.
Hmm.
Yeah, they would like to procure the germplasm, like and then do later parts by themselves.
Mhm.
So yes, and then I also received like calls from Argentina where they had the transgenics approved, like so for
Mhm.
wheat, like so they yeah they there is interest. Yeah.
Good, good. Okay. Any other question for Dr. Ruski? We still have a couple of minutes. To me, it's amazing the diversity you're assembling uh, through your gene editing and molecular means. I mean, it's amazing the diversity you are assembling. Okay, so it's a treasure there, I would say, not for just what you're doing right now, but for future research. Okay, so.
Thank you.
That's
Yeah.
great. Yeah. Well, looks like there's one more question that we can answer. Was there a trade-off to the lines that had allergen genes edited regarding protein contents, for example, in the genes, in the kernels?
Yes. So we have actually done like so protein content reduction we have not noticed because like we saw like there is some balancing effect that takes place.
Mhm.
And we have noticed in the past like it actually increased the content of globulins in wheat, and those are actually more balanced proteins, so it actually turned out to be relatively positive than we thought. Like it actually increased the lysine content of the wheat grains. So it made it more balanced because the gluten proteins are very imbalanced and they are
Mm,
lacking lysine, methionine, threonine, like so they got balanced relatively.
I
Like
see.
we didn't see like much reduction. However, there was some reduction in the seed size.
Oh, okay. All right. Well, thank you so much. Let me share the screen and okay. So thank you, Dr. Rushi, for your amazing presentation. As I mentioned before, the recordings to these presentations are going to show up tomorrow when we make the announcement for next seminar, which it will be delivered by Dr. Tom Purton from Pairwise about identification of a causal mutation of thornlessness in blackberry. It's next Thursday at 11, the same time, 11 time Eastern. Thank you so much for your attendance. I hope you have a great weekend, and we look forward to have you here with us in a week. Thank you so much, Sachin, okay? Appreciate it. Bye now.